Next speaker is from ThermoFisher Scientific, Dr. Omar Farah. His topic title is PSC spheroid differentiation into neural lineages.
Excellent. Can everybody hear me and see my slides?
Well, not in presentation mode.
Let me switch that up real quick, if I can figure it out. How about now?
Yes.
Excellent. Perfect. So first, I want to thank the organizers for hosting a great event. Honestly, I've been listening in all day, and the type of work everybody's doing is just really amazing. And thank you for giving me a chance to present today as well. My name is Omar. I lead our field application scientist regenerative medicine team at ThermoFisher. So anything related to stem cells, neurobiology, or even cell and gene therapy. But today I wanted to take a little bit of time and talk to you about new innovations when it comes to suspension culture and differentiating 3D spheroids into different lineages. We'll do a bit of a dive into neuronal differentiation as well. But before I start, I really want to take some time to just reflect, on the past several decades in terms of working with stem cells. I know 2023 will mark the 70th anniversary of the Charles Le Bon and Yves Clermont paper that came out in 1953 that identified spermatogonial stem cells, right? So I know a lot of us in the field tend to think about stem cells as relatively a newer and hot topic, and it definitely is, but the groundwork was laid way back when. It's going to be the DNA's 70th anniversary, but a lot of folks forget it's also going to be stem cells 70th anniversary, which I'm really proud to be part of that field. And as the field developed over the years, we have had quite a bit of struggles, I would say. I don't know if anybody would agree with me, but in terms of culturing the cells, getting them all characterized properly, engineering them, even figuring out differentiation protocols. But thankfully, with the great work everybody's doing, it's becoming easier and easier. And at Thermo, we really do pride ourselves in providing an end-to-end solution, really, for the workflow, the stem cell workflow in general that you see depicted here. So everything from reprogramming to culturing your cells, whether in 2D or 3D, characterization services and assays just used to non-biasedly quantify and qualify your cells, as well as tools to engineer them, right? CRISPR, TALENs, base editors now are really popular for the right reasons and really help us harness the regenerative power of stem cells. And then lastly, in terms of differentiation as well, protocols, kits that you want to use in order to highlight your or get the cell type that you want to be working with at the end. So in terms of culture of the cells, classically we've cultured stem cells in a 2D or monolayer culture, whether you're using Central 8, mTeaser, StemFlex. More recently, we've developed a media called the StemScale PSC suspension media system. And what this media system and protocol associated with it allows you to do is culture your pluripotent stem cells as PSC spheroids in their undifferentiated state, where they really allow you to scale up your cultures. With this media, you can expand your cells between 5 to 10-fold per passage over time. The workflows are pretty easy and it's consistent over time. Fortunately, I don't have a lot of time to talk about this media itself, because I do want to focus on the differentiation portion. But just real quick, I wanted to show you what these cultures look like. So these are 4 representative images that you see at the bottom right of 2 iPSC and 2 ESC cell lines that are cultured as pluripotent spheroids or pluripotent stem cell spheroids. And you can see that over time in culture, after you establish them, these spheroids continue to grow in a uniform manner. And around the day 4, day 5 mark, after initial establishment of these cells, they reach the 350 to 400 micron mark in size or diameter, where we recommend passaging them, which entails just simple dissociation followed by reestablishing of the cultures in order for you to avoid a hypoxic conditions or a necrotic core. But all in all, we've taken these cultures out, past 30 passages in suspension as spheroids. In the top right, I tried to squeeze in as some characterization in data where we show that whether you're looking at flow cytometric analysis of OCT4 or NANOG, or you're looking at global gene expression with our pluritest array, that these cells are indeed pluripotent, and they remain so for the entirety of that culture in suspension. We also have qualified these cells with our karyostat assay to make sure that they have no genomic abnormalities. And one thing I haven't shown here is we've also used our Oncomine assay, which looks at, which is a next generation sequencing assay that looks at point mutations in proto-oncogenes that are known to be commonly mutated in pluripotent stem cells. And thankfully, we have not seen any mutations throughout this culture. So at the end of your culture period here in suspension, you can get a very large number of cells, especially as folks are heading into more clinical translational applications that do really require a large number of cells for those applications. But you also end up having, or you have a high quality cells as well. And from this media itself, we started to ask, the question where I think the entire field is heading is, how do we use this in order to properly model disease, especially when it comes to the differentiation portion of our protocols. So we've done extensive work to actually generate a large number of protocols that you can take these PSC spheroids and in a complete 3D setting, differentiate them into different cell types, including cardiomyocytes, neural stem cell spheroids, definitive endoderm, as well as midbrain organoids, and I'm going to be talking about in just a second. And one of the kits that we had developed a while back was our GIPCO PSC dopaminergic neuron differentiation kit, which was originally designed, in a 2D setting, if you will, which allows you to take your pluripotent stem cells and differentiate them into a midbrain floor plate specific TH expressing dopaminergic neurons. And the 2D protocol for this kit is illustrated right over here. See if I can get my pointer going. There we go. Usually includes about a 10-day induction period, after which you'll have your floor plate progenitors. From day 10 to around day 21, you would expend them. And then from day 21 to about day 35 is when you would mature these progenitors to get TH expressing neurons. And like I said, this is usually the 2D protocol, but we wanted to see if we can adapt the media or even attempt to replicate the differentiation process, but completely in a 3D setting to better mimic what happens in vivo. And what we learned really is that for this kit, along with most of our other kits as well that we've used, is that this process is definitely doable. And in fact, it could be a little bit easier and more faithful to what happens in vivo once you differentiate in 3D. And what we did see is the fact that you can play around a little bit with the induction and expansion periods respectively. But by day 11, you would get the same number of floor plate progenitors as you would in a 2D protocol on day 21, saving you around 10 days in the process. And these cultures can be, by day 11, this is what they look like down here. And they stain positive for the right floor plate markers like FOXA2 and OTX2. And you could take these, spheroid progenitor spheroids at this point and plate them back down in 2D to get your dopaminergic neurons that express tyrosine hydroxylase, as you see over here, by day 21. Now, the next thing we wanted to do is, since we, for the first part, I showed you the maturation portion we were doing in 2D, we wanted to refine that to see how we could mature these progenitors in a completely 3D setting. So we started off with our PSC cultures that are on a rotating platform. And at the onset of differentiation, we learned that the size of the spheroid is really an important determinant for the success of the differentiation. So luckily for us, the stem scale suspension-based protocol or system maintains a homogeneous population of the spheroids when it comes to their size. So it's very easy to control when to start that differentiation. But by day 2, we started to observe some cavitation, as well as outgrowth and epithelial sheets, as you can see right over here. And by day 6, we were, these differentiating spheroids began to thicken and expand before we moved into the maturation portion. Now, for the maturation portion, like I mentioned, we tried a couple conditions, 2 of which that I'm going to show here, and then I'll follow up with another 2 in a later slide. But the first 2 were on a rotating, still on a rotating culture. So we're still maintaining these differentiating spheroids on an orbital shaker. And you can see here that from day 11 onwards, the floor plate progenitor spheroids were definitely cavitating. They did present with irregular shapes with the translucent outer layer. And they did at the end, whether you encapsulate them or you don't, they did end up expressing TH neurons that I'll show in a second. And these are representative stainings of, using our CX7 high content imaging system. What you see here is maximal projections of these stained organoids. And you can see here that for the rotating culture without the encapsulation, we were able to get some TH expressing MAP2 neurons. When we did encapsulate, we did get a better efficiency of deriving these neurons. As you can see over here, at a greater number of neurons that do express tyrosine hydroxylase. We are, however, very cognizant of the fact that the encapsulation process is not easy. It could be variable to a certain degree, and then dissociation and subsequent assays would also become very difficult. So we were trying to also move away from that. So instead of rotating cultures, the 2 other conditions we did test along the way were where we would mature these spheroids, but in a 96 well U bottom plate in static conditions, so without rotating them. And you could see here that in the static conditions in a 96 well U bottom plate, we were able to get a really high degree of MAP2 and TH positive neurons. The only thing with this particular condition was the irregularity that we saw between the different wells, or the different organoids in this case that were forming. And so for the last condition, what we decided to also include is to add some Geltrex into our media specifically. So we were not encapsulating the differentiating spheroids. We're actually adding 2% Geltrex to the media itself, just to give it a little bit more rigidity or increased viscosity in that sense. And what we did end up observing is a nice uniform rounded spheroid morphology for almost all of our differentiation, and a really high degree of TH expressing neurons that were derived as a result. And so the reason why we were kind of doing all of this and going on this mission really to optimize 3D differentiation protocols is for physiological relevancy, right? We often say that it's more physiologically relevant to do things in 3D, but in this case, we actually have some proof of that I'm very, very happy to share. As many of you know, the dopaminergic neurons in vivo develop in the substantia nigra, that upon cross-sectioning that part of the brain, you see some neuromelanin pigmentation in that particular region. And in 2D differentiation protocols, we never really saw that. While as with our 3D protocol, we started to see some of this pigmentation appear in our midbrain organoids as they were maturing into dopaminergic or TH expressing neurons. And this was true for the rotating culture to a certain degree, not very, very high, as you can see. We were able to improve this pigmentation, or the presentation of this pigmentation, with the encapsulation, when we encapsulated these differentiating organoids, as I referred to in the second condition. But where we did see the most impact is in our static culture conditions in those 96 well U bottom plates, where you could see here very, very clear and strong neuromelanin pigmentation, even at high culture periods, so day 63 in this case. And what I really wanted to kind of get across is the fact that now we can also start to do functional assays to really assess how well our model works compared to traditional 2D models that we used to use in the past. And so one thing we did was actually place these spheroids on an MEA, so a multi-electrode array, to be able to measure their electrophysiology. And as you can see here, that if we compare the raster plots and we quantify them in our 3D maturation protocol compared to a full 2D protocol, you'll see here that for mean firing rates, synchronicity, and number of active electrodes, the 3D differentiated cells always physiologically are more have more of an output or can fire strongly and more synchronously than we would see on our in our traditional 2D differentiation protocols. And then finally, what this really leads to is how do we use these models to disease model, right? I mean, we all know that dopaminergic neurons are one of the or the loss of dopaminergic neurons is one of the hallmarks of Parkinson's disease. So we internally have generated two different pluripotent stem cell lines that harbor SNP in either the SNCA gene or the LRRK2 gene, both of which are known to be associated with the Parkinson's pathology. And we're able to derive these lines, and I won't talk too much about the process here, but we're able to get these stable pluripotent lines that have these specific mutations. And what we did next was we used these two different lines alongside with a wild type control in order to differentiate them into these dopaminergic neuron organoids, and subjected them to different concentrations of rotenone, a non-selective inhibitor of the electron transport chain, to see whether the mutant or the wild type organoids were more or less sensitive or similar to each other. And you can see here that we do see an increase of apoptosis in the mutant organoids at lower concentration of rotenone compared to the wild type. Again, really, this is preliminary data that we're continuing to work on. But just to show that, in this 3D setting, we do actually have a bit of a case when it comes to better physiological relevancy compared to traditional models. And I know I've talked a lot about neuronal differentiation, but I really wanted to throw this in there as well, where we can do the same for other lineages. One example that I wanted to give here is we did take these pluripotent stem cell spheroids that you see in panel A, and we differentiated them into cardiomyocytes, or cardioids as we like to call them, in a 14-day differentiation protocol, where if this works correctly, I hope we'll just see this, we're able to generate within, within 14 days, these beating cardiomyocytes that one are amazing to look at, but also can be used for more physiologically relevant model creation, right? We are still in the preliminary phases of characterizing these organoids, but we do see that they express cardiomyocyte markers that are within the center of that organoid, enveloped by endothelial cells, and then fibroblasts as well, that all naturally differentiate in this case to make a mini heart, if you will. And so with that, I'll end it here. And just as a summary, I hope I was able to show you the fact that in a simple way you can use these PSC spheroids that we have data on how we can form them across multiple different cell lines consistently, but we can differentiate them into mature differentiated cell types in less time than the adherent cultures, and potentially in a more physiologically relevant manner. Now to end, I know you can go to thermofisher.com/STEMscale or thermofisher.com/STEMscale elearn. The link over here has a really nice e-learning module that highlights all of the data as well as the different factors in this culture that you can manipulate in order to customize it to your particular interest or protocol for the generation of these PSC spheroids. We also have a demo program for our orbital shaker. I know there's a table outside. I'm sorry I couldn't be there today, but there's a table outside if you want to ask one of the thermo folks about the demo program. I'm sure they'll put you in contact with one of our technical specialists or myself that can help arrange this demo where you get to try to form these spheroids without making the initial investment in the orbital shaker as well. And with that, I'll be happy to take any questions.
Thank you. Any questions?
Thanks for your talk. I have a question on the midbrain organoid. That have you ever analyzed the cell time composition at the later stage, like the day 70, by single cell RNA-seq or other approach? Especially, I'm wondering what's the percentage of TH positive DA neurons there?
That's an excellent question. This work is ongoing right now. We haven't had. I don't think we've done any single cell RNA work, but we are using traditional immunostainings just to be able to see what types of cells are present and delineating that before we start looking at the proportions of cells in there. The biggest kind of hindrance in this particular model is the fact that after a while the ECM composition becomes quite strong and rigid with these organoids, so their faithful dissociation without harming the cells is a little bit tricky, which is something that we're definitely working on to see if we can improve.
I'd like to ask, so for your cardiomyocyte differentiation in suspension, do you have to start with the stem cell spheroids, or you can start with adherent cells and do suspension for the differentiation portion?
That's a great question. We actually do have a kit for cardiomyocyte differentiation in 2D. I can send you the information about it. It's a pretty simple 2-media system protocol where you differentiate your PSCs into mesoderm fate, and then from the mesoderm we go into a cardiomyocyte into cardiomyocytes. So there is that option. The protocol that I have mentioned with our 3D spheroids is a little bit different. It actually, we're using shear to induce into, so Wnt activation to induce into a mesoderm fate, and then followed by in total inhibition of Wnt with the IWP2 in order to drive them from that state into a cardiomyocyte state. But we do have both options that I'm happy to share with you.
Right. I was asking whether you can start with adherent iPS cells and use the suspension protocol to differentiate cardiomyocyte spheroids.
Oh, I see what you mean. Typically, when you do it with adherent, they won't form the 3D structures. You will get a beating sheet, but maybe it's something that we can discuss, because that does seem interesting. Are you referring to dissociating these cells after they form cardiomyocytes and then forming your cardioid? Is that what you mean?
We can discuss this later. I do have another question about the so when you grow your iPS cells in suspension, so when we do it as adherent, depending on the iPS line, some we have to remove the spontaneously differentiated cells. We call it cleaning. Obviously, when you do it in spheres, you can't really do that. do you feel that when you grow them in 3D in suspension, you don't need to do that? No matter what line you try, they would stay undifferentiated?
That's a great question. I think it's even for the 2D, like depending on the media you use and how supportive it is, your degree of cleanup would be different, right? So with traditional medias, maybe the older, less lean medias like Essential 8, even potentially MT01, you'd have to clean up a little bit. With stem scale specifically in that 3D format, you don't necessarily have to because the cells are pretty well supported. That being said, once they do differentiate, or there is a degree of differentiation that for whatever reason something happens, your incubator oxygen levels or CO2 levels are not working that day or something like that, they're very clear and early signs that the cultures are differentiating. This is, we see cavitation pretty early within 24 hours, if so. And the really nice thing about our protocol is we always initiate our 3D spheroids from a single cell suspension as opposed to clumps. So you can easily redissociate the cultures, sort them if you need to, based on your typical TRA160 or SSEA4, and reestablish them with cells that you confidently know are only pluripotent. There are ways around it, whether it's morphological assessments or that, but we haven't seen that in all the lines that we've tested, and I think we have over 9 so far.
Cool. If there are no more questions, that's the end of today. I want to thank all the speakers and also thank you for sticking around here and online. We will see you tomorrow.
Thanks, everybody.